All right, welcome guys. So today we're going to be talking about PCA, t-SNE, autoencoders in the context of gene expression profiling. So first we're going to introduce what is gene expression analysis. Then we're going to talk about supervised versus unsupervised ways of analyzing these data. Then we're going to focus first on supervised learning, where we basically know the difference between two conditions, and we want to find differentially expressed genes between them. And then we're going to talk about unsupervised learning, and in particular, how do we embed this high dimensional data into lower dimensional space as a way to elicit the underlying structure. We're going to look at linear approaches for dimensionality reduction, including principal component analysis and singular value decomposition. And then we're going to look at non-linear approaches. And the example that we're going to take is t-SNE, or T-distributed Stochastic Network Embedding, which is one of the most popularly used methods, even though many others have since appeared. And then we're going to look at how we can actually carry out these lower dimensional non-linear embeddings using deep learning. And in particular, we're going to look at autoencoders. Everybody with me? This is like the last feedback that I'm going to be getting in class, where people get to sort of do facial expressions and wave at me and all of that. So what I'm going to try to do in the future, so basically for not next week, but 2 weeks from now, actually no, 3 weeks from now, is going to be to have a separate screen with all your faces and a separate screen with a lecture that you guys are going to be seeing. So I will be able to actually see you, and you will be able to ask questions. So basically, when you speak, I will have a separate monitor where I will be able to actually see your face, and I'll be able to see you nodding. So even though you guys are going to be spread throughout the world, or in your dorms or back home, depending on whether you're undergrads or grads, turn on your videos so that I know you're there, because it's very different to know that there's a human component. How many of you guys are actually leaving campus? Yeah, awesome. So main thing I want to tell you is don't panic. Basically, this is a rational approach. This is not a panic approach. If you look at what happened in China, they were able to contain the transmission because they isolated everyone. Italy is now undergoing this experiment. The entire northern Italy is sort of quarantined. We'll see if they're able to stop it. But Wuhan is actually going back to normality slowly. So there's experiments being carried out across the world on how to deal with this. Right now, I just want to tell you, you're all safe. Like, you're young, nothing's going to happen to you. The economy is changing, the world is changing, more isolation is going to be the norm, but hopefully we'll be able to sort of re-emerge from this and go back to normal. So don't panic, you're all safe, hug your loved ones after washing your hands, all of that. Anyway, great, so embeddings. So basically, first of all, biology, RNA-seq data. So as you know, DNA is encoded in sort of 23 linear strings in our cells, 2 copies of each of those, except for X and Y, which are slightly different. And then it produces this pre-mRNA. Eventually, this gets spliced into RNA, and RNA is exported from the nucleus to the cytoplasm, where it carries out all of its functions. And its main function for at least messenger RNA is being translated into protein, which then carries out all the functions of the cell. Everybody with me on this? So in order to understand what goes on inside the cell, one of the things that we can do is actually measure the level of RNA in a particular tissue, in a particular condition, in a particular cell line, in a particular cell. And sort of from that gene expression, infer the activities of the cell. So basically, there's a lot of high throughput ways of doing this. The primary one is RNA sequencing. The traditional one before that was microarrays. But basically, all of those are simply getting you counts of RNA molecules. Everybody with me? And those counts basically tell you what are the genes that are being expressed in any one condition. So all of the machinery that we've been talking about so far, with all of these networks to sort of learn the language of DNA and gene regulation and chromatin and all of that, the end goal of all of that is primarily to decide which genes are going to be expressed and which genes are not going to be expressed, and therefore which proteins are going to be made and which proteins are not going to be made, and therefore what the function of that cell will be. Everybody with me? So two technologies for figuring out RNA levels. One of them is hybridization of RNA into these microarrays, which basically have a spot for each of any kind of number of features of interest that we have, and then give you a number of molecules that hybridize to each of the spots. Everybody with me on this left part? Yeah? Any questions on this? Raise your hands if you have a hand. Gosh, I freaked everybody out. Can I get a show of hands for everybody who's still in the room with me? Awesome, good. This is reassuring. I'm not just hallucinating that there's a bunch of people watching. So who's with me on the left part? Right, super easy. So basically, we're generating a bunch of molecules that we're hybridizing to an array. And these molecules hybridize by complementarity. The more activity we have for a particular RNA, the more hybridization we're going to see. And the less activity we have for that RNA, the less hybridization we're going to see. So ultimately, we're going to get counts of molecules, or at least relative counts of molecules based on the hybridization intensity. RNA sequencing takes a different approach, which basically directly sequences the RNA molecules, now that sequencing is so cheap, and then we can sort of do a virtual mapping of those RNA sequencing reads into known gene models to basically count how many molecules did we see from every location of the genome, and therefore count how many mRNA molecules were likely generated from that location in the genome. Raise your hands if you're with me on the right part. Yeah, awesome. Good. So from both of them, we're basically ending up with some kind of gene expression matrix that basically tells you for every one of 20,000 genes in the human genome, what is the expression of that gene. And as I do this experiment over and over and over again for many different conditions, I end up with the expression profile of a gene, which basically tells me it was low in liver and it was high in neurons. Or when I was undergoing stress response, this gene was low, but then when I was back to quiescent state, this gene was high, okay? So these could be conditions, or this could be time course, or this could be different tissues, or this could be individual cells within a particular tissue. Everybody with me? So we can then do all kinds of cool analysis. We can basically take this N by M matrix and then ask which genes have similar expressions to each other, which conditions have similar expression vectors to each other, okay? So I can start clustering genes, or I can start clustering conditions based on their similarity. Is everybody with me? So this is the first type of analysis. I can basically ask about similarity between experiments. I can ask about similarity between genes. But this problem is extremely general. You could do this for epigenomic activity patterns. You could do this for neuronal firings. You could do that for patients and how much glucose they have in their blood versus how many lipids, etc. Okay, so this is an extremely general methodology. So basically what we're going to be looking at today is widely applicable beyond gene expression. So in general, we're going to have conditions on one dimension and then genes on the other dimension. And we're going to be able to define clusters completely de novo, basically say, oh, great, these genes appear to have similar expression across these conditions, and those conditions appear to have similar gene expression across the subset of genes, and so on and so forth. Yeah. Yeah.
One of the major difficulties I see when you see gene expression profile data.
Sorry, what's your question? What are the major difficulties?
Yeah, I mean, between the difficulties of using gene expression profile data as a training data set. One of the things that I think that is difficult is that gene expression profile data is a snapshot of a metabolic state.
Yeah.
Time point.
Yeah.
So for every condition measure at a different time point, you could have different results.
Yeah.
So how do you actually?
Yeah. So here's how to think about this. Basically, every time I measure something, when we're looking at single cell data, I'm basically measuring one cell. But that cell, I'm measuring it at a time, snapshot in time, okay? And then if I measured the exact same cell, which is not possible because after I've measured it, I've kind of killed it. If I measure the same cell, it would actually change from time to time. So when you have 1,000 cells, you might actually be able to put them in a pseudo time course that basically tells you, here's how every cell evolves through this, or changes through this cell expression space. Okay, so that's one way to think about this, but that's at the single cell level. So basically, even when I'm looking at individual cells, it's a snapshot in time. There's another thing that's happening, which is even at a given snapshot in time in that time course, what I'm doing is sampling. If I do RNA sequencing, I'm just catching RNA molecules and sequencing them. So there's two problems with sampling. The first is the RNA molecules that I'm sampling are being sampled from a larger set of RNA molecules. The second one is the reads that I'm reading are, again, some representation of these molecules. So basically, in the RNA extraction step, I'm sampling from the cell, and in the library construction, I'm sampling from that, from the extracted RNAs, and in my sequencing, I'm sampling from that library. So there's a lot of stochasticity in sort of which genes I'm going to capture. That's all at the single cell level, which is sort of, amazing that we're even there. But before that, most of the time we're looking at the expression of a tissue or a cell line, which is the average of many cells. So most gene expression data sets are going to be averages of many cells. So you should realize that we're averaging over time, we're averaging over the sampling, we're averaging over the stochasticity of expression of individual cells, we're averaging over individual cells of the same cell type, we're averaging over multiple cell types and cell states within a tissue. Is everybody with me? So basically, it's important to recognize just how many layers of complexity we're abstracting away when we're thinking of this gene expression matrix. But ultimately, after all that sampling, we're ending up with just a matrix of values that basically tells me what is the count of every gene in every condition. Is everybody with me here? Awesome. Again, all questions are interesting, so don't hold back, even when you're online. All right, so basically there's two ways to go about this. One is doing clustering to basically look for subsets of the data that make sense. The other one is classification to basically say, well, here's some known classes of genes. What are the expression patterns across conditions that allow me to classify these genes? Or here's two well-established conditions that I care about. What are the genes that are differentially expressed between those conditions? Everybody with me on sort of the difference between classification and clustering? So in clustering, you're learning the underlying structure of the data. And in classification, you're basically using some previous knowledge to distinguish genes or conditions. And you're basically trying to find the features that distinguish those conditions. So most of the time, we're going to be saying, okay, well, this person, so maybe I've sampled 100 individuals with Alzheimer's and 100 individuals without Alzheimer's. What are the genes that are differentially expressed between them? Okay, so that's sort of a feature learning on what distinguishes two previously known classes. Or these are known genes associated with, I don't know, liver function. Can I sort of find what are the expression profiles of those liver function genes that allow me to then go look for additional liver function genes? So basically working again on classification, but on the other dimension. Everybody with me so far? Okay, awesome. So what we're going to be doing today is basically focusing number one on classification. How do I actually answer the question of what genes are differentially expressed between Alzheimer's and non-Alzheimer's individuals? And then we're going to focus on clustering. Basically, how do I learn the underlying properties of my data? Okay, and the way that we're going to do clustering is dimensionality reduction. Basically, right now, I'm showing you the actual gene expression level across the actual conditions straight from the measurement. But what we're going to be doing is mapping those to lower dimensional spaces and studying the properties of those lower dimensional spaces. Sounds good? Awesome. So again, with clustering versus classification, objects are characterized by one or more features. In classification, we have labels for some of the points. For example, this is, I don't know, liver and this is heart. And heart expressed genes or heart function genes. And we have labels for some of the points. We're going to learn a rule that will accurately assign labels to new points. So for example, the subproblem might be, what are the features that are important? What are the genes that are differentially expressed. And the metric will be classification accuracy. With clustering, there's no labels. We're just going to be grouping points into clusters based on how near they are to each other. Or we might be visualizing the data to sort of gain insights as to what the actual underlying structure looks like. Okay? So let's dive right in. So basically, first, supervised learning. And in particular, how do I find the genes that are differentially expressed between Alzheimer's and non-Alzheimer's individuals? Okay. So basically what we're testing is there a difference in expression between, the samples that I've gotten from one condition and the samples that I've gotten from another condition? So basically, this might be the expression of 2 genes, or more typically 20,000 genes, but it's harder to plot 20,000 dimensional data. So basically, I'm going to be looking at 2 genes, and then based on everything that we're going to learn later today, those 2 dimensions could actually be the lower dimensional space that we're going to be projecting all these data into. Okay, so this might be principal component 1, principal component 2. So the question is, what changes? This is Alzheimer's, this is, normal, or this is normal, this is schizophrenia, and so on and so forth. So what we want to do is test whether the difference that we're seeing is actually significant, whether there is actually a significant gene expression change. So basically what we're going to be testing is the alternative hypothesis, H1, more expressive in terms of explaining the observed data than the original hypothesis. So basically every single time we're going to be looking for a differential expression, we're going to be doing a statistical test of significance. And in every such statistical test, you want to understand what are the degrees of freedom. How many genes am I looking at? How many principal components am I looking at? If I have 20,000 genes and I project them into, I don't know, 20 principal components, then effectively it's a 20-dimensional data set. So basically the degrees of freedom is actually changing, okay? So these are the 2 hypotheses. Basically, according to hypothesis number 1, the data sets are, so for example, distributed according to 2 independent dimensions. In hypothesis number 2, there might actually be some covariance between them. Okay, so this is one example of hypothesis. Or I might be asking, are they, and sort of, for example, you might see here what is the covariance here, and what you see is in this particular example, yes, indeed, there appears to be some correlation between the different dimensions, which basically supports hypothesis H1. So what we're going to be doing is basically a likelihood ratio statistic that is going to be asking what is the probability of observing all of my data according to hypothesis 1, and what is the probability of observing all of my data according to hypothesis 2. And then based on the ratio of these 2 probabilities, I'm going to be making a test that will basically say how much more likely is it that the data was in fact generating according to the alternate model. Everybody with me so far? Awesome. So to evaluate this p-value, we need to know what is the sampling distribution for the test statistic. Namely, what is that test statistic and how does it vary with the underlying hypothesis. And then we're going to be basically asking how many degrees of freedom did I have? And according to that, one very common test statistic is the chi-squared statistic, which basically depends on those degrees of freedom, and sort of the more degrees of freedom you have, the more stringent the score that you're going to be looking at is. And this is for a particular set of dimensions that we're going to be looking at. Okay. So then you want to know not only is it different, but what is the underlying distribution? And here there's a typo, obviously, so it's Poisson, not Poisson. Basically, what is the underlying distribution of the data? And very often we're going to be using, a statistic that will not be quite a perfect fit to the data, because of course the data was not generated according to our model. It was generated from nature by sampling. So you can ask, what is the goodness of the fit of the underlying distribution that I'm using to the data in order to actually choose the right distribution? Is it a Poisson statistic? Is it a negative binomial statistic, and so on and so forth? And according to the distribution of the data, you're going to be able to adjust the differential expression test accordingly. So DE-seq or differential expression analysis by sequencing is one extremely popular method that basically uses such a negative binomial distribution to basically test what are the genes that are actually differentially expressed between two conditions. So, you have a certain number of replicates, and then you're asking what is the sum of the expression of, or the distance between genes, summing over all the replicates, and what is the alternative expectation according to this negative binomial, according to the mean of the data, according to the standard deviation of the data, and so on and so forth. Okay. So you can basically use that to then ask what are the and the higher the expression with which you're measuring that you're measuring for the gene, basically the higher the expression of the gene, the more likely you are to find a significant change, because you have more opportunities for sampling. If the expression is very low, then you have very little chance of finding something significant. But as you get to more and more confidence in the number of molecules that you've measured with higher mean expression of that gene, then if you find a log-fold change, for example, that's, I don't know, a two-fold change or a three-fold change, then that is much more likely to be significant because you're measuring more molecules effectively. So if I only sample 3 molecules in one condition and 1 molecule in the other condition, chances are that this is just statistical fluke because I wasn't measuring sufficiently deeply. And that basically means that even though these are showing very high log-fold change, because their average expression is so weak, I just don't have significance. But as I go to higher and higher average expression, then the number, then the amount of change that I can detect as significant is getting more and more precise. So I can detect subtler and subtler changes with more genes. Raise your hands if you're with me on this. Okay, awesome. So this is a very standard analysis that is basically telling you which points are significant in red, what is the differential activity in Y, and what is the mean expression in X, which basically allows you to sort of infer these. And you can ask what is the differential expression of a set of genes. And one approach is the hypergeometric distribution that Professor Gifford showed you a few weeks ago. That basically it says how many genes for that particular gene set were differentially expressed, how many were not differentially expressed, and how many would I have expected by chance given the overall size of sets A and B. And this is basically simply adding up anything that's greater than this counting distribution of what are all the ways of sampling N2 from a total number of endpoints, all the ways of sampling K elements from a subset of N1 points, and of course, N minus 2 minus K, N2 minus K, which is the remaining elements sampled from the remaining of that set. Okay, and that's sort of what you saw already. So we're not going to spend too much time on that. And of course, because you're testing 20,000 genes every time, you have to penalize for the number of tests that you're carrying out. So basically what Bonferroni correction does is that it basically says, if I do 100 tests and I want a p-value of 0.05, then I'm going to divide that by the number of 100 tests. And this is very typical in genome-wide association studies where we have, 6 million SNPs on average that we're testing based on their independence of what we were talking about last lecture. And then you have to correct for that by effectively dividing by 1 million different hypotheses. And then the same thing happens for gene expression. So as Professor Gifford mentioned, there's a lot of problems with Bonferroni. So there's sort of less stringent tests that are basically sort of doing a progressive hardening of your, or softening of your threshold. Yes, after you found the first one, it's kind of like Bonferroni, but as you go further, it sort of loosens out. Okay, so this is the supervised part. This is basically how do we test for differential expression between two conditions for finding the set of genes that are known to be differentially that are discovered to be differentially expressed between two known conditions that are different. So basically, I've sampled Alzheimer's and non-Alzheimer's individuals. I want to find what are the genes that are differentially expressed. Everybody with me? Good. So now let's switch to unsupervised learning. So basically what we're going to be doing now is simply visualizing the data in a bunch of different ways, testing different approaches of exploring the data, and then generating hypotheses which we can then go and test statistically. Okay, so it's a very different kind of approach. It's when you don't know the right answer, when you don't have a very well-defined problem that you're starting with. Okay, so the particular approach that we're going to do to unsupervised learning now is dimensionality reduction. And there's many uses of this dimensionality reduction. One of them is data visualization. I mean, I always say that the rule number one of genomics is look at the darn data. Okay, just look at it, because you can, from looking at it, discover all kinds of things that you might not realize. You might be running the world's most awesome statistics, and it turns out that the data was just, an error, or it was just all, basically that entire chunks of a chromosome are missing, and so on and so forth. So it's very important for data visualization. It's also important for data reduction. Basically, when we were talking earlier about gene 1 and gene 2, well, I'd rather just do PC1 and PC2 and sort of look for ways of reducing the overall complexity of the data. It can be very helpful for classification after the fact. So you can basically first do dimensionality reduction and then classification. You can use it to look for trends. You can use it to look for what are the underlying factors that are governing the dimensionality of my data set. For example, the factors might correspond to all of the different processes that are going on inside the cell. So if I carry out 100 different gene expression experiments, and then I sort of reduce the dimensionality of the data, I can then ask what are the subsets of genes that are driving the overall variation that I see, even though there might not be any gene labels, and there might not be any condition labels. I might just be able to ask what are the subsets of genes that tend to move together, and what are the subset of conditions that tend to sort of group together. And that can be helpful for recognizing biologically meaningful gene sets. For example, this whole pathway appears to be going up and down in a coordinated fashion. It's by reducing the dimensionality of my data that I was able to recognize this underlying factor driving this. Raise your hands if you're with me. Awesome. All right, and then it can also be very useful for noise reduction. Basically, when I measure 20,000 genes across 100 conditions, the underlying dimensionality of the data is not necessarily 2 million. It's actually much, much smaller. And the reason for that is because of that coordinated, gene regulatory circuitry. Basically, genes don't just turn on and turn off independently. They turn on and they turn off in response to all of these gene regulatory circuitry that we talked about the last few weeks. So in order to reveal the true underlying data, I might actually have to reduce the high frequency components of my data that basically are more likely to represent noise. In other words, if I see that this entire group of genes going up and this entire group of genes going down, and this entire group of genes going up again, but individual genes within it show little variation, then that little variation is most likely to be noise. So dimensionality reduction allows me to actually reduce the stochasticity, the randomness of the data, and sort of reveal just the biologically meaningful sort of major drivers of the variation. Is everybody with me? Awesome. This is actually, a double-edged sword, because sometimes the major axis of variation are the noise. For example, your batch, like the batch at which you did the experiment, might actually be driving a big chunk of variation. So in some cases, we actually remove the first few principal components, and then we remove the last few principal components, and sort of we focus on that middle of the distribution. And then some of the questions you can ask is how many unique subsets are in the sample? You don't need a classification. You don't need labels for that. You might say, well, there appears to be 3 clusters here. How are they similar or different? What are the underlying factors that influence these samples? Which temporal trends are correlated or anti-correlated? What measurements are needed to actually differentiate between these subsets? And how do we represent the stuff that's interesting about the data? Or which subset is this new sample coming from, even though we may not have labels for these subsets? Everybody with me? So the traditional, so basically the approach that we're going to, that is going to be sort of overarching throughout all of these different solutions that we're going to see is the embedding of these data in a lower dimensional manifold. Okay, so a manifold is basically a lower dimensional topological space where locally it kind of acts like a Euclidean space. Okay, this is very abstract, but it's basically telling you that even though I'm observing data in 3 dimensions, the underlying data comes from a 2-dimensional surface embedded in these 3 dimensions. Okay, so for example, if I'm I don't know, somewhere on Earth, most of the time I just need the XY coordinate instead of the XYZ coordinate in 3 dimensions. So basically what I get from the satellite GPS data is a place in 3D space where I am. But in order for us humans to best understand this, projecting that 3D coordinate in the solar system space onto the 2D coordinates of latitude and longitude on Earth is actually a much more natural representation of my 3D coordinate in space. So in the same way, if we take these 20,000 genes by 100 different conditions data, it's extremely sparse. It's extremely, out there in terms of the dimensionality of the space. But we could project all that down and basically say, oh, well, this is one of the cancer conditions, and this is one of the, I don't know, proteasome genes. And then that gives me a lower dimensional space within which I can actually interpret the data much more uniformly. Raise your hands if you are with me on this one. Awesome. Any questions so far? All right. So manifold embedding is basically a structure that preserves the mapping of this high dimensional space into this lower dimensional space. And the manifold learning basically learns this lower dimensional space that enables such a manifold embedding. So basically we want to both embed our data into this lower dimensional manifold and also learn the shape and the dimensionality of this lower dimensional manifold. Okay? So that's what we're gonna start doing. And one way to do that is principal component analysis. What is principal component analysis? It basically says, well, I have a bunch of, I don't know, blood and urine measurements from 65 people, 32 non-alcoholics, 33 alcoholics. I want to sort of ask, well, yes, the data has all these different dimensions. So this is a trivariate plot that basically tells me here's the actual raw data in each of the dimensions, but I might have 50 of these dimensions. Plotting the data in 50 dimensions can be hard. Visualizing the differences between them in 50 dimensions can be hard. But what I could do is actually project the data onto the first components of variation. So basically, what are the largest components of variation? So we have a population of points that is measured on some random variables. And these random variables represent the axis of a Cartesian coordinate space within which the population resides. And our goal is to develop a set of other axes that are linear combinations of the original axis in the directions of greatest variability. So that's what principal components analysis does. As the name suggests, it's basically the components that are sort of capturing the principal variation. Okay? So how do I do that? Basically, the idea is that I'm going to basically learn what are these principal components variation and then project onto them. And how do I do, and after the projection, I can, instead of looking at X and Y, I can actually look at PC1 and PC2, which is taking all of the same points and then projecting them onto that space. Okay? And then PC1 might be capturing most of the variation, PC2 might be capturing less of the variation, and that's reflected in the overall spread of the points. Okay? So how do I do that? One way to envision it is start with a one-dimensional space example. So basically, we're going to sort of first look at a very degenerate space and then sort of move up to the higher dimensions. So in one-dimensional space, this is basically equivalent to mapping a set of two-dimensional points onto a one-dimensional line in a way that minimizes the difference, the deviation of all of these points from that line. And the reason for that is that, it sort of captures the major axis of variation by sort of minimizing all of these distances. Does everybody see why? So But this is a very simple example. It's going to one dimension. In general, we're going to be going from n dimensions to k dimensions, and n could be, I don't know, 10,000, and k could be 20. So we're going to have to develop some methodology for doing this systematically, okay? And the methodology comes from linear algebra. Basically, what we're going to be doing is learning eigenvectors and eigenvalues. And very simply, an eigenvector of a matrix is a vector such that multiplying that matrix and that vector together effectively gives me a linear scaling of that vector. So the way to think about that is that you have, you're basically trying to find the transformations that capture identity along that direction. So the way to think about that is to think about your matrix of points as a transformation of that space, and then these vectors being the axis and the dimensions along which that transformation is linear. So eigenvalues and eigenvectors are very helpful for symmetric matrices, because, and so we can capture these orthogonal eigenvectors and orthogonal eigenvalues, and all these eigenvalues are basically real, and these eigenvalues of these positive semi-definite matrices, which are defined like this, are basically all non-negative. All of these should be reviewed. And basically, what eigendecomposition does is effectively transform any matrix of expression values, again, 10,000 genes by 100 conditions, into a set of principal vectors of variation that basically tell me, here's how every gene, sort of basically, this is the major pathways. And maybe I only have 20 major pathways. Even though I have 10,000 genes, maybe I can summarize them into the 20 principal axes of variation, the 20 principal factors. And the way to write that in linear algebra is to basically say that I'm going to be taking a set of scalars that basically tell me what is the overall magnitude along each of these dimensions of variability, and a set of vectors that basically tell me how is the expression of that gene looking like across, or of that eigen gene looking like across all of these different conditions, and then just multiplying these across. Okay, so it's basically a typical matrix diagonalization problem. So basically the columns of U are the eigenvectors, and then the diagonal elements of this diagonal matrix, where all of the off-diagonal elements are 0, are effectively the eigenvalues that basically tell me how much variation is there across each of the dimensions. Raise your hands if you're with me on this. Awesome, very cool. All right, so basically every time we have a symmetric matrix, there exists a unique eigendecomposition where Q is orthogonal, and the transpose of the matrix is actually the inverse of the matrix, and then the columns are normalized eigenvectors. But I don't always have these symmetric matrices. I don't always have diagonal matrices. So that's where singular value decomposition comes in, where for any m by n matrix of some rank R, there exists a factorization that allows me to now have two slightly different vectors, U and V transpose, that basically allow me to express the orthogonal elements, the orthogonal eigenvectors of this matrix. And the way that I'm going to be building this is that because I want to decompose this matrix A, I'm actually going to be looking at A transpose and A transpose A, which are going to be n by n matrices. Okay, so these are going to be actually having eigenvectors and eigenvalues. So with an m by n matrix, I'm instead going to be computing the eigenvectors, the orthogonal eigenvectors of A transpose, and these are going to be U. The orthogonal eigenvectors of A transpose A, which are going to be V transpose, and then the eigenvalues of A transpose are going to be the eigenvalues of A transpose A's, and they're going to be basically the square roots of these eigenvalues, and these are going to be the singular values. So what singular value decomposition does is that it does some acrobatics here to basically transform this matrix into effectively identical dimension parts that allows me to now start decomposing even non-symmetric and non-diagonal matrices. Everybody with me here? Awesome. So there's a geometric interpretation of singular value decomposition, which is that instead of basically looking at this whole matrix M, which is effectively a shearing matrix that basically transforms the space into, you know, both a rotation and scaling. And the singular values and the singular vectors are going to be, you know, sigma 1 and sigma 2 here, basically the principal axis of variation. So instead, what I'm going to be looking at is, you know, V star, sigma, and then U. And then the sigma is just multiplying things by a different scalar along each of the dimensions. And then V star and U are going to be simply a rotation and another rotation. Okay, so that's sort of the geometric interpretation of singular value decomposition. Instead of sort of combining these operations together in this sort of super high dimensional space, I'm going to be splitting them into something that rotates without scaling, something that scales along the different dimensions, and then something that rotates back. Raise your hands if you're with me on this one. Awesome. Awesome. So basically this is, you know, one way to illustrate it, to basically say that even though A is M by N, U is M, you know, N by N, and VT is M by M, and the singular values are the ones along the diagonal, and then this is the rank R, and then I ran out of rank, so I'm kind of, you know, left with no additional dimensions here. Okay, so the true dimensionality only goes down to here. So yeah, here's an example. And the reason why this is so cool is because I can actually use that to achieve low rank approximations of my overall data set. So when I have these 10,000 by, you know, 100 conditions, I can basically find a low rank approximation by effectively trimming the singular value and just using a subset of those. Okay. So what this allows me to do is basically find an optimal low rank approximation of my matrix A with rank K that basically minimizes the difference between this original matrix A and my you know, approximation X, given this Frobenius norm, which is basically the sum of squared differences for each of those. Okay, so basically as I'm doing this, so A, K, and X are both M by N matrices, and typically we want the lower rank approximation to be much smaller than the rank R. Is everybody with me? What's really cool is that I can prove that the optimal such approximation is nothing but the first eigen, or the first singular values of the singular value decomposition, where I've just set all of the other values to 0. Okay, so I have the same U here, which I get from A transpose eigenvectors, the same V here, which I get from A transpose A eigenvectors, and then all of the singular values, which you can obtain from these eigenvalues. And then I just zero out the smallest ones, and then that gives me actually the exact minimization. Raise your hands if you're with me here. Awesome. Raise your hands if you think this is kind of cool. Yeah, awesome. It's extremely powerful. So linear algebra is awesome, and you should really embrace it. So basically the way that you do principal component analysis is that you find the eigenvectors through singular value decomposition. You decompose S into these orthogonal rotation matrix, these diagonal scaling matrix, and then you basically find the columns of this matrix, and then you just zero out the lower rank approximation. Okay, so when I do this on the MNIST data set, which is super high dimensional, this is what I get, right? I basically have this principal axis of variation that basically tells me, you know, along the first two principal components, I can decompose on one side the digit 1, and on the opposite side the digit 0. On one side up here, the digit 7, and then on the opposite side, the digit 3. Okay, so this is very cool. It basically says that I can visualize the first two dimensions of this very high dimensional space, and already I can see a separation of digits. So that's pretty awesome. And then this is just looking at PC1, PC2. I could then look at PC3, PC4, PC5, PC6, or PC2, PC3, and so on and so forth. And then I will find additional separations of my data set along each of these dimensions. Raise your hands if you're 100% with me so far. Awesome, great. So this is kind of great. This is kind of awesome. But again, visualizing the first 10 principal components can be cumbersome because I have to iterate through all of these pairwise plots to basically see, are the digits really separable? Okay. So what we're going to look at next is how we could actually get an awesome separation of all of the digits into their own little clusters, completely de novo. Okay. So the next thing that we're going to look at is how do we embed into these lower dimensional spaces in non-linear fashions? Because what PCA and SVD do is a linear reduction in this dimensionality. But again, the whole point of this class is to go into the sort of non-linearity ways of understanding biology and complexity and health and disease and so on and so forth. So let's now dive right into non-linear reductions of dimensionality. Everybody with me so far? Awesome. So I think you guys deserve a stretch. So everybody stand up. It's the last chance I actually get to see you stretching. Reach for the sky. Reach to the right, from the left, and reach to the left, from the right. And then hop. 6 feet. 6 feet. All right, do we have blood flowing to your brains again? Because we're gonna need it. All right, so who has heard of t-SNE before? Raise your hands. Awesome. Who knows how it works? Awesome. Good. 0 hands up. All right. So let's talk about non-linear embeddings. So we now have this expression matrix, and what we have is, I don't know, maybe 10,000 genes across 100 conditions. So we have a 100-dimensional vector where I have individual genes on this 100-dimensional vector. And I'd like to identify what are the clusters of genes. I want to do some unsupervised learning through embedding of this very high dimensional space on a lower dimensional space. Okay. Another example, along the condition dimension, I might have 100 different experiments, each of which is measured in a 10,000 dimensional space of, I don't know, 10,000 genes. Okay. So now my dimensionality is 10,000, and I have 100 data points, each of which is a condition. Okay, everybody with me on those two examples? The third example is individual cells. I basically look at a human brain. I've measured 5,000 cells from that human brain. And I want to know for every cell, I have an expression of 10,000 genes in some space. I want to know what is the distance relationship of all of those 5,000 cells. A realistic example, in my lab, we've now sequenced 2.3 million single cells from 1,000 human brains. We'd like to create a representation of what are all these 2.3 million cells looking like, okay, in a 10,000 dimensional space. So, PCA is not quite as satisfying, and sort of looking at all of these different dimensions, each pairwise, sort of understand sort of what are the major subclusters is, very difficult. So that's where t-SNE comes in. This is, that's where this t-statistic distributed stochastic network embedding comes in. So what is stochastic network embedding? Stochastic means that there's some probability associated with it. Embedding means that I'm going to take this high dimensional data and sort of project into a lower dimensional space. And then network basically means that I'm going to be looking at the pairwise relationships, the edges effectively, between every point and every other point in this kind of dimensional space. Everybody with me here? So that's what t-SNE actually means. So how are we going to do that? What are the things that I want to preserve? I want to preserve the distance so that when 2 points are close in this high dimensional space, they continue to be close in this lower dimensional space. And I want to preserve the neighbors. Basically, if this is my closest point, then I still want this to be my closest point. Even though I'm going to go from a 10,000 dimensional space into a two-dimensional space or a three-dimensional space. Everybody with me on what we're trying to achieve? So the way that we're going to be doing it is by preserving the neighborhood. So for example, in this particular case, this red point is near these three blue points. In this particular case, this red point is not near these three blue points. So I've kind of broken that neighborhood. So I don't want that. What I want is to actually preserve the neighborhood. Even though I'm mapping from this high dimensional space to this low dimensional space, I want to sort of preserve the overall relationships between these points. So how am I going to do that? The way that we're going to do that is we're going to be asking, how do I map a distribution of distances into a different lower dimensional distribution of distances such that the probability of finding point xj as a neighbor of point xi is basically preserved in this mapping of point xj into point yj in the lower dimensional space, and the mapping of this point xi into point yi in this lower dimensional space. Okay, so I'm going to be transforming these points. Every one of those points has 10,000 dimensions, and every one of those points has 2 dimensions. Okay, so basically I'm going from a 10,000 dimensional space to a 2-dimensional space. So I'm mapping these points to those points. Basically, these points have lots of coordinates. These points have only 2 coordinates. And I'm mapping them in a way that preserves the distribution of probabilities for neighborhoods. Okay, so the probability of observing point J given that I'm focused at point I and looking at my neighborhood is effectively going to be this exponential function. So in stochastic network embedding, it's basically just a Gaussian function. And for t-distributed stochastic network embedding, it's going to be a slightly different distribution. It's going to be a t-statistic instead of Gaussian. Okay, so very subtle difference. But that is going to allow me to now effectively map these distributions to each other. So it's going to be a stochastic search for a lower dimensional space that preserves that property. Have a comment?
Yeah, just the T distribution, it's still Gaussian in high dimension, but it becomes T in low dimension.
Yeah. So basically, I'm going to be mapping it into a T distribution in this lower dimension. Okay. So I'm going to be setting a bandwidth surrounding my points such that this conditional here has a fixed perplexity. Okay. And the perplexity you can think of as the effective number of neighbors of my point. Okay. So the way to do that is that basically what you're actually doing is setting the resolution, the radius over which you're going to be looking for neighbors according to some parameter. So I can basically choose to look at only 5 neighbors, only 30 neighbors, only 50 neighbors, and so on and so forth. And that's a tunable parameter. Okay, so that's the perplexity, which is effectively the Shannon entropy of this PI distribution. And then, you know, the original paper basically recommends values between 5 and 50. Okay, everybody with me here? So basically, I'm going to be looking at this Gaussian distributed distance matrix. I'm going to be mapping it into this t-distributed, distribution. And then I'm going to be preserving the relationships between points, focusing in on the local properties each time. So I'm not going to be caring about preserving the relation between this point and a faraway point at all. The only thing I'm going to be caring about is, can I preserve the local similarities? Raise your hands if you're with me here. Awesome. Great. So we want to choose an embedding that minimizes the divergence between the low and the high dimension similarities. So I'm going to be looking at the similarity of the data points in this high dimension here. I'm going to be looking at the similarity of the data points in this low dimension here. And this is a t-statistic. This is a Gaussian. And then I'm going to be matching them in a way that sampling centered on I and finding its neighbors J is going to be matching that distribution. Okay, so how do I calculate the deviation between distributions? So the typical way of doing that is VD dimension, basically. So, you know, or sorry, the KL divergence. So basically, I'm going to be asking what is the KL divergence between these two distributions, and then minimizing that. Everybody with me so far? So basically, the original distribution is some kind of Gaussian center in every point, and then the mapping distribution is this student t-statistic in red. Okay. So what we're going to be doing is basically using a gradient method to find such an embedding. So basically, I'm going to be using a cost function, which is this KL divergence between the P distribution and the Q distribution. The P distribution is the original, the Q distribution is the original high dimensionality distribution. And then the P distribution is the new lower dimensionality distribution. And that's going to be basically telling me not about the overall distribution of points, but the local distribution of distances centered on each point with all of its neighbors. Everybody with me? So KL divergence is an asymmetric function. So KL of P versus Q is actually very different than Q versus P. And in this particular case, because P is actually featured on the numerator here, as opposed to the denominator and in the product, we're going to be looking at a large penalty when the original distance is large, and now I've brought 2 points that were initially far away closer. So I really don't want to do that. It's not okay to bring distant points closer. But a small penalty if I have an original small distance and I now have spread them a little further apart. Okay, so it's okay to move points further from their not super close neighbors, but you have to preserve the super close neighbors. Okay. So what t-SNE does is it mainly preserves the local similarity structure of the data. And that this gradient is simply going to be, you know, sort of taking the derivative of that thing relative to yi. And I'm basically going to be optimizing over this space. Okay, so it's going to be an iterative approach. And at every iteration, I'm going to be optimizing that gradient and then mapping that space into the original high dimensional space into a low dimensional space. Okay. So basically, these are the original gradients. So you can see this low dimensional distance here versus the high dimensional distance, and you know, sort of how this is actually changing over time. Okay. So now, I don't know if you guys remember the PC1 versus PC2 of these characters. What t-SNE allows you to do is effectively map into a space where the super proximal points are still super proximal to each other. But, you know, if 2 points were kind of far from each other, now they can be pushed as far from each other as we want. Okay. And you can see here just how beautifully this separates digits from each other. And you can see here that 9 and 4, for example, have a lot of similarity. 3 and 5 have a lot of similarity. 5 and 6 have a lot of similarity. And 1 is very different from 6 and so on and so forth. Okay, everybody with me? So the main thing that you should take away from this is that locally, if you look at any 2 points that are near each other in t-SNE, they must have been near each other in the original plot. But any points that are far away from each other could have actually still been very close to each other. Okay, but not super, super close. Raise your hands if you're with me here. Yeah, awesome. Who feels that they've learned something important? Awesome, good. Question?
Good question. I'm just wondering what is similarity to this from here?
So you're basically asking, can I sample points? Basically, what is the probability with which I will sample Pj as a neighbor of Pi? Does that make sense? So that's what that sampling is. That distance is simply a probabilistic measure of how likely am I to choose this particular point as my neighbor. Does that answer your question?
Yeah, no, I was just, maybe it was very simple than that. I was just wondering, like, 9 and 4 in... Oh,
what do I mean by similarity in this particular example? Well, when humans confuse them, they're more likely to confuse a 9 into a 4. I don't know if, you know, you've asked somebody their phone number in a bar and you're like, oh, is that a 4 or 9? Anyway, I don't know if you guys still do that. No, you just swipe right, swipe left. Anyway, so but you know, if your grandma basically says, "Hey, go, grandpa, this is the number," she writes a 9 instead of a 4. You know, this is sort of how humans confuse them. Basically, the only difference between a 9 and a 4 is sort of the relative length of these numbers, and the difference between a 6 and a 5. And my daughter is still drawing backwards epsilons instead of 3s. And, so anyway, it's kind of cute to sort of see your kids learning how to write digits and the mistakes that they make and the sort of differences, and it kind of matches this distribution here. So what's the difference between 5 and 3? It's just like this thing here. And again, my daughter kind of confuses those sometimes. And then what's the difference here? You just close that loop. So it kind of makes sense that you're, you know, embedding into this space. Yeah.
Question on the MS data. So if we thought about the vector pixel values, we want those separate dimensions.
Exactly. So basically, we've taken I don't even ever remember. So what's the dimensionality of the pixels? 156. Sorry, 20 by 20. So basically, we have 400 dimensional vectors, and then you're mapping them onto this 2-dimensional space. So again, we went from 400 to 2, and suddenly it preserves a lot of that similarity. And then all of these weird color points in the middle are just points that were probably mislabeled. No, I mean, correctly labeled, but will probably be mislabeled by most, you know, algorithms because they just look like each other. Yeah?
You would like to, that's right. With the PCA, you can actually, you know, the ways of stream to the PCA, you can have the data by saying, gene A is important to separate the data, but
sorry, here?
No, in general, like with PCA, you know, like for every gene, there's a way for the PCA, right?
Ah, yeah.
So then you can know which gene is the most important to whatever.
Yeah. So I don't think that this applies to t-SNE. Basically, it's you kind of lose any kind of interpretation. Like the x-axis means nothing. No. After the fact, you can do that. After the fact, you can kind of say, okay, great, let's define these as clusters. And now what are the genes that separate these clusters? So that's something that we do a lot. So basically in single cell data, for example, you create all these clusters and then you sort of say, okay, now that I have my clusters, what are the genes that are differentially expressed? And then you can go back to section 2 of the lecture to basically search for statistically significantly differently expressed genes. Does that answer your question? Any other questions? Awesome. Who thinks this is kind of cool? Awesome. So t-SNE is extremely popular, and the reason is that it kind of works like magic. It sort of takes these weird things and sort of progressively pushes them away from each other. So let's explore a little bit of that magic. Let's like play with some of the t-SNE parameters. So we talked about the this neighborhood size, where we were basically asking how many local neighbors do I want to include in my search. So this, the size of this circle. In this particular case, the perplexity, if you wish, is kind of like 2. It basically says I'm only going to care about 2 neighbors at a time. But I can tune that perplexity and recommend values between 5 and 50. Okay, let's see how this affects the results. So we can actually tune this perplexity parameter and basically ask, what do I get? So here's the original data, basically a bunch of blue points and a bunch of orange points sampled from, I don't know, 2 Gaussian distributions of equal spread, equal standard deviation. And then I can play with perplexity and sort of scale it from only looking at 2 neighbors at a time to looking at only 5 neighbors at a time, 30 neighbors at a time, 50 neighbors at a time, 100 neighbors at a time. Okay, what do I get here? And why do I get that? With 2 neighbors, you can kind of see why this is happening. Because when I'm looking at only 2 neighbors, well, it seems that these points are way close to each other, and these other points are way close to each other, and these other points are way close to each other. So it's going to create a space that captures these local similarities, but perhaps over captures the fact that, these guys were actually separated from those guys. And it kind of spreads the data into perhaps way too many clusters. Raise your hands if you can see that. Awesome. And as I start increasing that perplexity, I now have 5 neighbors that I'm considering. Again, this is an approximation. That's not exactly what it means, but it's a good way of thinking about it. Then I can kind of see the local structure starting to be a little more uniform here. And I'm kind of capturing that all of these points are near each other and all of those points are near each other, even though I'm still finding some substructure with everybody with me. And then as I continue to increase the complexity to 30, then I see that, wow, all of these points have exactly the same 30 neighbors, so I'm going to lump them all together. Okay? And as I start increasing it to 50, then I'm starting to spread the points out a little bit more because I'm now starting to learn about maybe some other neighbors here that I'm trying. So instead of having super dense clusters, I have to slightly more spread out. And then when I increase my perplexity to 100, suddenly I have all of the points being local neighbors, and there's no more separation between the clusters. Raise your hands if you like get it. Awesome, great. So there's no magic here. Basically, the way that this behaves is exactly the way that you would expect it to behave. You should just not interpret that in any way. The only thing that matters is the local proximity. And the layout, so many people misread the layout to actually mean something. Or, oh wow, this is far from that. No, that doesn't mean anything in this thing. The only thing that matters is the local proximity. Completely. Any other questions? Yeah. Yeah. You can think of it that way. Basically, you can basically say, yes, indeed, maybe that's not exactly the right approach. Okay. Things are showing enough, and you don't really care about outliers much. Any other questions? Awesome. So, what about the number of steps? Basically, sometimes you will create your t-SNE plot and you will see, oh wow, the data is kind of pinched. 5 more steps. And then the data collapsed completely. Try more steps. Oh wow, the data is spreading and so forth. Okay, so the number of steps actually matters. Like you will actually get some intuition, start recognizing sort of not enough iteration kind of t-SNE. So again, this is an iterative procedure. It sort of follows this gradient with a just in space. So you would expect it to be different from one point to the other. Even though it actually shows dramatic robustness very quickly. The other thing to realize is even if your data is dramatically different from one cluster to the next cluster, is this Gaussian has a spread that's tenfold greater than that Gaussian, the t-SNE plot couldn't care less. And the reason for that is that it only looks at the local neighbor relationships. Okay, so it adjusts the metric by design. So by design, it adapts to your variation. And oh gosh, I lost this. You use an algorithm that's designed to do this. It seeks to make local variation and where you are in the place. This is an extremely dense. It uses a much more refined measure of distance. In places that are very spread apart, it uses a much broader distance. That's what it does here. So therefore, it sort of shows you local relationship by caring about the role distance. Everybody with me here? Awesome. The other thing is you, again, by design, only care about the local distances. And if these two clusters are near each other and further from that cluster, that's not what t-SNE was made to look for. Very often you're going to see them as equidistant. that's okay. And you know, it's not an issue. Sometimes we capture and it's whole relationships further out as well. Okay, everybody with me? And then you also should not read too much into the data. So basically, this data is actually random. And you can see here that with very, very low perplexity, you can see some structures. And this is, again, by design, if you understand the underlying math, this is just basically trying to capture what are the pairwise groups of points. And, you know, as the data is sparse enough in sampling from your Gaussian, and as the perplexity is small enough, you might actually start again, if you look at perplexity values between 5 and 50, and optimal of the slide, which is in perplexity value, you can see that, oh yeah, it kind of does. Okay? And then, you know, some relationships are not always preserved. You can kind of see this, but again, highlighting this central zone here of perplexity values. And at a very similar consistency here. See that we need, you know, different information for relationship between these points, but at very low complexity, it doesn't necessarily need to factor that. And you can see this is taking and then running. Anyway, you can try this at home. It basically shows you that you can capture that proximity quite well. All right, we'll try to put these videos on there. All right, everybody with me on this thing? Oh. Yeah, just a small comment. Do you want to come in here? No, come, because this way people will hear you better. Yeah.
So we just went through what's non-parametric t-SNE. So on the problem set 3, we're actually going to look at parametric t-SNE, because with this method, you have new data points. The method relies fully on just transferring the data points you already have, so you can't put new data onto your t-SNE. So using neural nets and parametric t-SNE, we can actually bring in new data. So that's problem set 3.
So this has been basically looking at linear embeddings, looking at non-linear embeddings. Well, what does deep learning, you know, allow you to do? Well, what deep learning allows you to do is actually create some of those non-linear embeddings using these convolutional. How? By tricking what is by nature a supervised algorithm into an unsupervised algorithm. Okay? So the idea is that I have an input, which is the exact same cat, and I'm trying to predict cat from cat. Sounds easier, right? Well, it's easy unless you blindfold the algorithm, which is basically the bottleneck here. It basically says, oh, sure, I'm going to give you a 1,000 by 1,000 picture, and you're going to have to predict a 1,000 by 1,000 picture. But in the middle, you're only going to have 2 neurons or 2 perceptrons or 2 units. Everybody with me? So basically, one way of using these deep neural networks that we've learned about is by copying the output to the input, but dramatically reducing the number of layers that actually forces the algorithm to learn effectively representations in lower dimensions of this super high dimensional data. Because you're trying to predict the super high dimensional data from these lower representations. So here's the distribution here. You think it's like, yeah, the really cool idea is basically, you know, sort of allowing you to do dimensionality reduction in non-linear lower dimensional embeddings using just a trick on infrastructure methodologies that we've already seen. It's Autoencoders, and that's same thing. Okay, so auto basically means that this is the same. And encoder basically means that you're finding lower dimensional representation because you're forced to reuse parameters. Okay. All right, so basically what you can do is learn, you know, a function that minimizes the difference between sort of first this encoding function and this decoding function, you know, okay? And you can actually use that to effectively recover signal that's corrupted by noise. So basically, if you have a bunch of noisy data and measurements, you can actually feed it into an autoencoder with some loss function. Basically, store the original learn manifolds, these lower dimensional manifolds, input 9 and output 9, even though I'm sort of going through such a bubble. Okay. So here's the autoencoder learning of this MNIST data. So basically, you can now, you know, scroll through here and basically see how the decoded widget changes as it goes through different parts of this. So you can kind of see that, you know, I'm sort of moving 7s here, and 7s are starting to look more and more like 1s. And then, you know, sort of I'm going through the 1s, and then I get to the 9s, and get to the 8s. And you can continue as you go through. Okay. And then this is basically using 700 nodes here, 300 nodes here, 300 nodes here. So basically 3 layers of, and poof, opens the bottom of only 2 nodes and 300, 300, 400. Okay. So this is basically state nodes in the middle. Then I try to predict these digits from these digits. Here's the actual along this dimension. This is something. Yeah, awesome. Yeah. But that middle bottleneck layer is basically capturing a bunch of nonlinear relationships between the regional but effectively you're ending up with only 2 dimensions. Sounds good? All right. So what did we learn? We talked about gene expression analysis. We talked about 2 ways of addressing supervised clustering versus unsupervised classification. For supervised, and then for unsupervised, we looked at embedding into lower dimensional space. Linear reduction of dimensionality to the components and similar values. And then we looked at nonlinear embeddings using distributed stochastic network embedding. Locally tuning that distance and then spreading points apart while maintaining the local similarity function. And we looked at how we can do that with deep learning, of clamping down the complexity of the model through these intermediate layers with very few nodes, and then clamping that out. Last question? Yeah. Clustering is unsupervised. Oh, do they have a typo? Yeah, sorry. Clustering versus supervised. Any other comments? Awesome. Guys, traveling in this current, state of affairs is hard. So basically, wash your hands. Don't let's see. Don't panic. Wipe every surface as you sit on the airplane if you're traveling home, and then wash your hands before going to the plane. Wash your hands before getting out. Wash the belts before putting it on. Wash the windows before touching them with your disinfecting wipes. So I had the little like plastic bag of disinfecting wipes as I was traveling, so you can use that. And then when you get home, wash heavily, and then hug mom and dad really, really hard. Okay, because you're all gonna be fine. You are not the danger group, but you might be spreading it to people who are at risk. So please minimize interactions as much as possible. And good luck. I'll see you guys on video in 2 weeks. Thank you all. Awesome.